and welcome to Attempting Science. My name is Irma, and I'm from Leo iGEM team. And I'm here today with a friend from another team.
Hi, I'm Jacob, and I'm from the Chalmers iGEM team, and I'm here to talk about my project.
So, let's begin. What's your project called, or what's your main idea for the project?
So our idea is to treat colorectal cancer using an engineered yeast. So we basically have three parts of our project, and that is detection, monitoring, and treatment. And we want to be able then to make a pill that anyone with colorectal cancer could eat, and that pill would then contain yeast, which would attach itself to colorectal cancer cells and be able to secrete some anti-cancer molecules, which would then break down the cancer. And we will also want to monitor this process. So our idea is that these yeast cells would produce some kind of gas vesicles, little gas bubbles that you can see with ultrasound.
Cool.
Yeah.
And how is it going so far?
So far, it's going all right. The whole gas vesicles part has not gone that well because it's like around 12 genes that required to make these bubbles, and that's all a bit complex and many things that can go wrong. And so far, we haven't had much success with that part, and it's getting to the end of the summer, so we're planning on perhaps dropping that part while keeping our eyes open for other possibilities in that department. But for the whole detection and treatment part, we're making some progress. We have gotten some results, and we're doing some further testing to see what exactly has happened with our yeast and if it's doing what we think it should be doing.
Yeah. And how would this affect your project if you lose one of the three parts?
I mean, that's it's quite sad, of course, because it's like some part that we thought would be really cool that hasn't really worked out. But the whole monitoring part is not one of the crucial components of this project. It's more the treatment and detection. And even though it didn't work out, it was like a cool concept.
Very cool.
So we're thinking that maybe one could try this concept with something else, and like if we had more time, maybe we can find out what went wrong. And we'll also like look at other possible solutions if for monitoring when this doesn't work.
Yeah. Do you have any ideas what the other monitoring suggestions could be?
Yeah, we had like some talk with with a doctor from I think she was from Malmö, and he said that maybe one could try to add some kind of component that's slightly radioactive.
Cool.
That not in doses that would be harmful at all, but enough that it could be monitored with equipment. Because apparently our idea with the gas vesicles was also not that stable in the beginning, because even if one would be able to make these gas vesicles, the whole ultrasound detection is a bit not that accurate because you have a lot of other gases in your gut and the colon, which would disturb the image. Yeah, we're thinking about something like that, or maybe some other kind of marker molecule, perhaps some colored pigments, or that you could see in your excrements, or something like that.
Cool. And what would your project mean for the world if you succeed?
Yeah, that's a good question. We're thinking that since colorectal cancer is one of the most harder forms of cancer to treat, it's like I think that's the third most lethal one, standing for about 10% of the deaths. And the problem with just colorectal cancer is that it has a very high chance of recurrence. So even if you treat it, there's always going to be some that survive, and therefore it's good to attack the cancer from different angles. So you could combine this with the normal like chemotherapy. So the chemotherapy kill some of the cells, but not all of them, because some of them might be a bit resistant or something. But then those cells might not be as resistant to our method of attack.
Mm. And if we go into the two parts that you seem to be succeeding with, what exactly are you doing?
Yeah, so the whole detection part is normal gut cells in your gut. They have this kind of protein called sorry, I don't remember the name, but
That's okay.
they have a protein on their surface, and most of the time there's not that much of it, and it's kind of like hidden at the surface, not really able, you're not really able to get to it. But cancer cells in your gut, they, for some reason, they express this protein a lot more, so there's a lot more of it on that surface, and it kind of stands out a bit, so it's much easier to get to. And there is a Streptococcus strain that has developed a protein called HLPA, called for human-like protein A, and this protein can attach to that. The syndican-1 was the protein that's expressed on the surface of the gut cells. So this human-like protein A can attach to syndican-1. And so we're thinking of trying to express this human-like protein A in our yeast cells. So they would be attached to some kind of anchor in the membrane. So then when the yeast flows by the cancer cells, then they would attach to the syndican-1 and kind of get stuck there. And then we reach the other part and the treatment part. So we're thinking of when enough yeast cells have gathered at the cancer cells, they would secrete these signal molecules through something called quorum sensing or pheromone signaling. Cool. And that's basically normally used for mating between different yeast types. But we made it so that they can detect these signal molecules, and when they reach high enough concentration, because there's a lot then of our yeast cells in one small area at these cancer points, at a certain threshold they would start to secrete some anti-cancer molecules, where we have two candidates. We have one anti-cancer molecule called P28, which basically is a small peptide that can just penetrate the membrane and go straight into the nucleus. And there it will interact with a gene called P53, which is responsible for the cell cycle. And this gene is very often damaged in cancer cells, because that's like what's enabling the cancer cells to divide with unlimited amounts and stuff like that. So nearly all cancer cells have some kind of problem with this gene. And the P28 somehow interacts with this gene, and it has shown that it has anti-cancer properties.
Cool.
And another candidate for an anti-cancer molecule that we have is myrosinase, which is an enzyme that can degrade a certain thing that is very abundant in certain vegetables, such like broccoli or stuff like that. So when you eat this and you have the enzyme, they will degrade a certain molecule in the broccoli and turn that into an anti-cancer molecule.
The degradation of broccoli, or?
So the broccoli has something, I think it's called glycosonolate or glycosonol. And this is then degraded by the myrosinase into another certain molecule that is shown to have anti-cancer properties.
Cool. That sounds amazing.
Yeah.
And when do you think this could be on the market, and when could it be tried?
Oh yeah, that's a very hard question. Normally these iGEM projects, they are more like proof of concepts. And we're basically trying to see if this kind of works in a lab environment, and if we then would have been successful with all of this and been able to show that, hey, this actually works, then starts a really hard and long process of actually making sure that it works efficiently and that it's not harmful, and there's a lot of steps on the way in order to actually turn this into an actual pharmaceutical. And that process usually takes many years.
Yeah, absolutely. And just succeeding with your project is a huge step on the way.
Exactly.
Yeah. And how do you think your team is gonna? How do you think? Oh, this. How do you think the competition is gonna go for your team?
Yeah, I think it will turn out fine.
Yeah.
Because I think our idea is quite cool and has a lot of potential.
Yeah.
And so far, like most of the stuff seems to go quite well.
Yeah.
And if we look at what you need to do in order to get like a gold medal or whatever, we seem to be going quite strong in that. We just have to produce to characterize one of our parts and make sure that it works somehow,
Yeah.
and send it in, and that would probably be enough for a bronze medal, because like you need to actually create a part that works. But all of the rest of the steps in order to like reach silver and gold, stuff like human practices and how we can make our progress more incorporated into society and stuff like that. That's we're doing great progress in.
Yeah. And now the big question: why do you use yeast instead of E. coli?
Yeah. So that's a good question that many people have asked us, because usually this type of stuff is much more easier in E. coli, and most of the genes that we use in this project are from prokaryotic bacteria, which then would make much more sense to put in E. coli, which itself is a prokaryote, instead of putting in yeast, which is in eukaryotes. But the division that we're under, called systems biology, has made a lot of research into yeast.
Yeah.
So if we work with yeast, we can use their expertise in a much better way than if we would have worked with E. coli. And also we thought it would be a challenge to ourselves, because all of these parts has already been shown to work in E. coli, not together but by themselves. So we thought that it's not really that interesting to just take some already working parts and put them together and call that project. We thought it would be cool if we could actually make it work in eukaryotes as well. And then for that we had to try some kind of new methods, because for example the gas vesicle genes, in prokaryotes you can transcribe a lot of different genes at the same time. You don't need to stop between each gene, whereas in eukaryotes you have to do that. So we had to introduce something called 2A peptides between each genes, which then would cleave off itself, so that in that way we could produce a lot of different genes at the same time in a eukaryote, exactly like a prokaryote. And then when this protein would be created, it would break off into each of the individual genes.
Okay, cool.
Yeah, but we don't know if that actually worked, because we didn't succeed with that part.
No. How was it? How was the process of coming up with this idea?
That was a fun process. We basically looked a lot on partly older iGEM projects, and then we looked a bit at what kind of problems are in the world, and we got some papers from our supervisors with for example the 2A peptides. That idea came from them, and we spewed out a lot of different ideas in the beginning, and then we kind of made sort them into themes. So we therapeutics, monitoring, environmental projects, stuff like that, and we kind of looked at what seems to be the most popular subjects, and then we focused on those few subjects, and we took a weekend where we went to a little cabin in the woods or at an island, and we just basically sat and brainstormed different ideas, and in the end we kind of came up with this, and it was not really that we came up with all of this at the same time. It's that we put a lot of different parts from our previous ideas together into one, and so that maybe all of these parts can work together in a nice project.
Cool. Would you like to say something more to the podcast?
I mean, I'm really happy to be here and to talk with you guys, and hopefully our project will do well, and hopefully your project will do well as well. And I'm looking forward to the jamboree and seeing how it all will go.
Sounds great. Then we'll see you in Boston. Thank you.
Thank you.
Bye.